Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Researchers at the University of Gothenburg in Sweden, together with their colleagues at the Barcelona Beta Research Centre in Spain, the University Medical Center in Ljubljana, the scientists developed new tools to detect these subtle changes and confirm their results in human samples. At a time when the incidence and social costs of dementia and Alzheimer's disease in particular continue to rise, this breakthrough is very timely as it could enable the detection of. The findings are also important for the testing of therapies against this devastating disease. Alzheimer's disease is characterized by two pathological changes in brain tissue. For tau, individual units of the protein can aggregate into finely ordered fibrillar structures facilitated by a biochemical process called phosphorylation. Throughout the disease process, amyloid beta and phosphorylated tau, p-tau, are released from the brain into cerebrospinal fluid. The amount of the released proteins are used as reliable surrogate markers for clinical diagnoses of Alzheimer's disease. This makes it difficult to identify people with the disease at the very early stages before it is too late. How can we, therefore, reliably detect these subthreshold disease changes? To address these challenges, the scientists discovered that there are specific forms of p-tau that undergo very minute increases in cerebrospinal fluid and blood in people with emerging Alzheimer pathology. Consequently, the researchers developed highly sensitive techniques to measure these biological markers that precede clinical signs by several years. In the first study, conducted in the alpha parent cohort study at the Barcelona Beta Research Centre, BBRC, with the support of La Caixa Foundation, about a third of the 381 people evaluated had brain evidence of Alzheimer pathology but without any cognitive problems, meaning that these changes. Remarkably, the new p-tau markers correctly identified these emerging abnormalities measured in cerebrospinal fluid and regular blood samples. Subsequent studies performed in Gothenburg, Paris and Ljubljana revealed that these new markers continued to increase from the preclinical stage through the onset of cognitive problems to the late dementia stages. For this reason, progressive increases in p-tau could provide insights into the biological and clinical development of Alzheimer's disease. The studies are now published in the leading journals in biomolecular medicine and Alzheimer's dementia, the Journal of the Alzheimer's Association. Candidate drug trials have not been too successful. He added that, the practical challenge, however, is that these very tiny initial changes are incredibly difficult to measure. This compromises our chances to identify and recruit preclinical AD patients for clinical trials. Dr. Thomas Karikari, an assistant professor at the University of Gothenburg, who co-led the discovery, commented, "The remarkable findings reported in these publications show that the new highly sensitive. These tools therefore have the potential to advance population screening and clinical trials." According to Dr. Mark Suarez Calvet, neurologist and ERC researcher at BBRC, the biomarker detected in blood may change clinical practice in the coming years, since it will improve the diagnosis of PAT. Note: content may be edited for style and length. Please support our channel to grow by pressing subscribe button as well as the bell icon for daily science updates. Thank you.